Exhibiting a difference in charges in a given area is called blank. As with blank charge on the inside of a neuron, okay, I don't know at all. Most cells are negatively charged on the inside, whereas the outside, the extracellular space, is positively charged. Exhibiting a difference in charges in a given area is known as polarity. Okay, so the inside has a negative charge, the outside has a positive charge, and the difference is polarity. An example of this is the polarity in a water molecule where there are negatively and positively charged regions. See chapter 2. Neurons and muscle cells are known as excitable because they can respond to stimuli. That is, unlike most other cells, they have the ability to rapidly and dramatically change the internal charge, sending it well into the positive range. This sets off a cascade of events, allowing neurons to communicate with each other. Okay, let's look at the question. Okay, so polarity is in the first box. Polarity. Then negative and positive. Okay. All right. Next question. The most numerous glial cells are blank, involved in regulating ions and nutrients in neurons. I'm not sure, but I think it might be microglia, but let's look it up. I think I remember this before. Neuroglia, nerve glue, is a collective term for 6 types of cells, 4 in the CNS, 2 in the PNS. They are also called glial cells and have quite diverse functions. They include protection, insulation, and general support. Glial cells far outnumber the neurons in the CNS and are of the following types. Microglia are small spider-shaped cells scattered throughout the central nervous system. They develop from white blood cells called monocytes. Far outnumber the neurons in the CNS. Okay. They also help to form scars in areas of damage. Okay. Scroll down a little bit. I think it's glial, but okay, there's our picture. We did that before. Microglia, okay. Astrocytes are the most numerous of all glial cells and can make up 90% of all the cells in certain regions of the brain. Preventing capillary leakage, they play a major role in the maintenance of the blood-brain barrier, including the regulation of ions and nutrients in neurons. That's astrocytes. Okay, I'm glad I looked it up. Go ahead and go back to the question. It's astrocytes. Astrocytes. You just read that. Okay, next question. An inactive neuron contains proteins and ions that provide a blank charge when the cell is at rest. Okay, I don't know that we've read that. Let's read it. Okay. A charge in a cell is known as a membrane potential. The term indicates that there is the potential for charges to move across the cell membrane. When charges flow from one area to another, they create an electrical current. The resting membrane potential is the neuron. In a neuron is about negative 70 micromillivolts. That is when the neuron is at rest or inactive. Compared to the outlets in your house that carry a potential of 110 volts, this is very small, but it is quite a big deal to a neuron. So, okay, it looks like it's negatively charged. Let's go down and make sure. But it looks like there's a negative in front of it, so it's probably a negative charge. Let's read a little more and see what it says. Charges in cells arise mostly from ions. The potassium K plus ions are found in higher concentration on the inside of a neuron. However, the overall charge on the inside is due to many of the negatively charged proteins and large phosphate and sulfate ions. They are known as fixed because they cannot cross the membrane. Okay, so okay, let's go back. I think it's at rest, it's negative, I think. Yeah, negative charge. Because it should be positive, but the bigger proteins and the ions are making it negative, is what we read. A negative charge. Okay. Among the cells of the nervous system, mature blank lose the ability to divide, but blank can continue to divide and produce new cells. Okay, let's read about it. See what it says. Okay, we read that part. We read about monocytes and areas of, okay, keep going. Okay. Epididymal cells form oligodendrocytes are along the myelin sheath. Glial cells resemble neurons, but do not generate or conduct impulses. Most glial cells can still divide, making more cells. Most mature neurons lack centrioles that are needed for cell division and therefore lose this ability. Okay. So, okay, so um, um, most mature neurons lack it, but glial cells can. Let's go back to the question. Mature neurons lose the ability to divide, but go back to the, I thought it said glia. Glia can continue. Is that what we thought we saw? Let's go back. Glial cells, whoops, it was up on the top. Oh no, yeah, keep going under the picture. Sorry, my mistake. No, under the picture. I was mistaken. Glial cells resemble most glial cells can still divide. I don't know what that second word is because it only has one box. Go back to the question. Mature neurons, but try glia, Glia. I don't think that's what they want, but there's only one box, so we'll have to see because I think it's supposed to be glial cells. But try that. Oh, see, they would have taken two words, but there's one box. Okay, so it's neuroglia or glial cells or okay. Okay, so we'll know for next time when they ask you. Myelination is provided by blank, and the PNS, this insulation is produced by blank. Okay, that's in the same section. I didn't read you about the myelin sheaths, but let's read about that. So keep going. It's under the okay, so oligodendrocytes are aligned along the axis of neurons. They produce insulating layers of myelin called a myelin sheath, the oligodendrocytes. This sheath increases the speed of conduction of neurons in the brain and spinal cord. So oligodendrocytes are the ones that produce it. And scroll down. And Schwann cells produce the myelin sheath in the PNS that surrounds the axons of peripheral nerves. Neurolemma. Okay, when peripheral nerves are damaged, their axons may regenerate. The neurolemma plays an important role. In contrast, CNS and axons are myelinated by oligodendrocytes. CNS, okay. So, okay, let's go back to the question. Okay, so CNS nerves are done by the oligodendrocytes. And in the PNS, this insulation is produced by Schwann cells. So it's the last one. Yeah, okay. Voltage gated sodium channels open when the what? Okay, voltage gated sodium channels. Let's read about it and see what it says. Okay, at this point, voltage gated sodium channels open. So let me read a little bit about action. So a neuron will stay at rest until it is stimulated. A stimulus is anything that can change the resting potential of a negative 70 millivolt in either direction. The key to understanding How this happens is the action potential, which is a rapid change in the membrane potential, first in a positive direction, then in a negative direction, returning to the resting membrane potential. An excitatory stimulus will open chemically gated sodium channels. Based on this concentration gradient, sodium ions flow in, making the neuron less negative. A special type of excitatory stimulus, the threshold stimulus, usually a specific neurotransmitter, will cause sodium to flow in long enough to change the potential to negative 55 millivolts. This is called the threshold potential. The first part of an action potential is the change from a negative to a positive charge. This is known as depolarization because the inside and outside of the neuron now each possess a positive charge. Okay, let's so the voltage gates open when an excitatory stimulus happens with sodium channels. So let's take a look at the question again. Okay, so voltage gated sodium ions open when the I would say the neuron's membrane reaches threshold potential. Because we didn't read anything about potassium. We read about sodium, but not potassium. Yeah, I think it's that one. Yeah. It reaches the threshold. It has to reach threshold in order to open. Neuroglia that provide a protective coat around the bodies of peripheral neurons are called. Okay, so if it's in the peripheral nervous system, this is Schwann cells. I don't remember. It's like SCHWANN, I think. I think that's the spelling. Capital S. Oh, capital S, because it's somebody's name. So CH WANN. Schwann cells. Oh, neuroglia that provide. Okay, let me read about it because I thought it was Schwann cells that because they're going to ask us again. Let me read about the satellite cells because it's going to ask us about that later.
I nailed it. Getting kids geared up. $40 set after 6. I've been using UV hard gel, and of course, I'm back with my girl set. There's a difference between a nail tech brush.
There's nothing on the screen except the question. A neuronal membrane is said to be polarized when the inside of the membrane being negatively. I don't think so, but let's read about it. Polarization go up. Okay. Okay, so a difference in charge when there's from the inside to the outside. Okay, go ahead and go back to the question. Okay, hold on. Neuronal, I'm gonna look this up another way. Let's see. And when the inside has a negative charge compared to the outside. That's true. That's true. That's what we read in the, because the inside is negative and it becomes positive after the sodium ions coming to it. Okay, next question. Opening of sodium channels in a neuron results in opening of the sodium channels in the membrane of a neuron. Okay, hold on. Opening of the sodium channels. A positive charge inside the membrane and depolarization. So it would be the last one, depolarization. It's not going to be polarized anymore. Okay. At rest, the interior of a neuron has a negative charge in the presence of select all that apply. Okay, they told us. I don't remember. I think I remember proteins, but let's read about it because there's more than one answer to this. Okay. Let's see. Keep going. Go up a little bit. No, I mean, go down the page. Scroll up, yeah. Okay, yeah. So charges arise from ions. Potassium ions are found in higher concentration on the inside. However, the overall charge on the inside is due to many negatively charged proteins and large phosphate and sulfate ions. So negatively charged proteins and large phosphate and sulfate anions. So let's go back to the question. Negatively charged proteins and phosphate and sulfate anions. The third one. Okay, next question. An action potential at the blank zone of an axon causes an electrical current to flow to the adjacent region of the axon membrane. Okay, let's look at because I don't think we've read this far into it. This is about an action potential at the blank zone. Okay. An action potential or impulse at the trigger zone of an axon. So it's trigger zone, causes an electric current to flow to the adjacent region of the axon membrane. It's trigger. Okay. Nice. Both neurons and glial cells can continue to divide after reaching maturity. No, that's false. Neurons don't. Glial cells can, but neurons usually don't. I would say that's false. Okay. Impulse conduction, a series of blank blank, occurs along the axon from a trigger zone to the axon terminal. Let's read about it. Oops. Okay. Okay. So this local current stimulates the adjacent axon membrane to its threshold and triggers another action potential. The new action potential in turn stimulates the next adjacent region of the axon. As this pattern repeats, a series of action potentials occurs along the axon to the axon terminal. So it's action potentials. Action potentials is what they want. Action potential. Okay, let me get it. Give me a minute. All right, what's the next question? Which of the following occurs when a threshold potential is reached? I believe that's sodium channels open. Because we read about that. Let's read about it, but I think that's what we read. So At this point, yeah, sodium channels open because the threshold stimulus is reached, and then at this point, gated sodium channels open, and the charge rises to about negative 30 millivolts. Yeah, we did read that. So sodium channels open at that point. Okay. Let's see. Myself on. Alrighty, so now it's saltatory conduction is confined to the blank regions of myelinated neurons. I don't know. Let's read about it and see what it tells us. Okay. Okay, so this is conduction. We don't scroll under the picture. And action potential movement along an unmyelinated axon is called continuous conduction because it flows uninterrupted along its entire length. A myelinated axon functions differently because myelin insulates and prevents almost all ion movement through the axon membrane it encloses. The myelin sheath would prevent impulse conduction altogether if the sheath were continuous. However, nodes of Ranvier interrupt the sheath. Action potentials occur at these nodes where the exposed axon membrane has sodium and potassium channels. In this case, the adjacent membrane is brought to threshold at the next node down the axon. An impulse traveling along a myelinated axon thus appears to jump from node to node, eventually to the axon terminal. This is known as saltatory conduction. Saltation is Latin for jump or leap. It is many times faster than conduction on an unmyelinated axon. Saltatory conduction occurs on myelinated axons in both the PNS and the CNS. So it occurs at nodes of Ranvier. Okay, let's go to the question and see. To the, oh, okay, let me see. Let me see. Saltatory conduction is confined to the, let's see what it says. Saltatory conduction. Oh, is that what it wants to do? To the blank regions. To the regions. Um, Trinodal. That's the adjective form of that. Nodal, nodal regions of myelinated neurons. Al, Nodal, nodal. Um, yeah, try it. Because it doesn't have room for run VA, so I'm interested to see. Yeah, node or nodal or unmyelinated. Okay, they would have taken either one. Okay. Which neuroglia provide a protective coat around the cells of peripheral neurons? Okay, let's read about this. So we're looking for the type of neuroglia that do that around the cell bodies of peripheral neurons. Let's read the, okay, keep going. It's going to be down below the picture on the list of stuff. Okay, so including the regulation of ions in between. Epididymal cells, glial cells resemble neurons that do not. Schwann cells provide, okay, Schwann cells provide the myelin sheath that surrounds the axons of peripheral nerves. So Schwann cells would, okay, go to the question. So Schwann cells provide the protective coat around the cell bodies of peripheral nerve. Schwann cell. No? Why does it say satellite cells? I didn't see satellite cells anywhere in the, okay, we're gonna have to, okay. A protective coat around, okay. Let's go on and see. Let's see what the next question is. I didn't see satellite cells anywhere in the, but okay. Which occurs to cause depolarization of a membrane. Okay, so hold on. The opening of sodium ion channels. So it's when sodium ion channels open, okay, and so the sodium ion channels open and sodium flows into the cell. It should be the second one. Sodium ion channels open and sodium diffuses into the cell because it becomes more positively charged to balance it out. Remember, it's negatively charged, so the positive ions have to flow in to balance it. Because the cell at that point is negatively charged. So when the positive ions flow into it, it won't be polar anymore. If it flows out, then that's when it becomes polarized. Okay. But this is depolarized. This is when it doesn't have a charge. Okay, let's look at what's the next question say. Regarding the speed of nerve signal conduction, conduction is faster in myelinated neurons than in unmyelinated neurons. Speed of conduction is the same, or conduction is faster in unmyelinated neurons than in myelinated neurons. Okay, so how does conduction speed compare between myelinated I think yeah, so myelinated neurons do travel much faster. That's the purpose of myelination. That's one of them. So the first one, conduction is faster in myelinated neurons than an unmyelinated neuron. Uh-huh. Okay, cool. An action potential at a trigger zone of an axon causes an electric current to flow to the adjacent, okay, an action potential at the trigger zone of an axon. Okay, hold on. Causes an electric current to flow to the membrane of an adjacent. Okay. Okay, let me see what the answers are. Okay, let's look at it. I think it's the last one, but let's look at the reading. I want to see what it says. Um, okay, so an action potential causes an electrical current to flow to the adjacent region of the axon membrane. The adjacent region of the axon membrane. The new action potential stimulates the new adjacent region of the, okay, so, okay, let's go back to the question. The adjacent region of the axon membrane. So they're using the first one, to the adjacent region of the axon membrane. I think they're talking about the initial part of it. Go ahead. I think they're talking about the initial trigger that it has. Okay, let's continue. An impulse on an axon with a blank diameter would travel faster than one on an axon with a blank diameter. Okay. An impulse on an axon with a diameter. Larger conduction of impulse in axons with larger diameters is seen. They can conduct so larger diameter axons conduct faster. So that would mean that, yeah, an impulse on an axon with a large diameter would travel faster than an axon on a small one. So it's the first one. Okay, yeah. An action potential propagates as, okay. Let's see. A continuous wave. I don't think she had that, does she? As as Let me see. I'm not sure. Let's look it up. I'm not sure if it's an action potential propagates as. Okay. An action potential movement along an unmyelinated axon is called continuous conduction because it flows along its entire length. Okay. An impulse, okay, that's saltatory conduction. Keep going. The speed of an impulse is proportional to the diameter of an axon. The greater its diameter, the faster the impulse. Okay. Following an action potential, a threshold trigger will not trigger another action potential on that portion. This brief portion, called the refractory period, limits the frequency of action potentials and also ensures that the impulses progress only in one direction down the axon. This is because the area upstream from where the action potential has just occurred is still in the refractory period from the previous action potential. Okay. But that's not telling me what. Okay, keep going. This is all the. Mm, mm. Okay, let me see. What is the an action potential propagates as and positively charged. Mm, Let me see if this tells me anything. Give me a second.
Welcome to 2 Minute Neuroscience, where I simplistically.
Let me see if this gives us anything that we can use. I'm not sure because the book doesn't really make sense to me here. Let me see. Let's go back to your question. So an action potential propagates as positively charged ions rush into the axon, making the axon more, I think, I think it's the second one. I think it's the second one. Mhm. The functional connection between neurons is called a blink. Okay. Okay, let's see. The functional connection between. Let's look it up, but I think that would be a synapse. I don't know that we've seen this word before, but I think that's what they're talking about. Let's read it. Yeah, it's a synapse. The neurons at a synapse are not in direct physical contact, but are separated by a gap called the synaptic cleft. Okay, synapse is the word we want there. Okay. And then we have in myelinated axons. In myelinated axons, impulses travel from node to node. That's saltatory conduction. Saltatory. A-L T-A-T-O-R-Y. Mm-hmm. At a synapse, the neuron that releases the neurotransmitter sends the signal is the blank, and the neuron that receives the signal is the blank. Okay, let's look at the book and see. Okay. Okay, so we have to scroll past the picture. When you get a text message, the person texting is the sender and you are the receiver. Similarly, the neuron conducting the impulse to the synapse is the sender or the presynaptic neuron. The neuron that receives is the receiver or the postsynaptic neuron. The mechanism whereby this message crosses the synaptic cleft is called synaptic transmission. So let's go to the question. So it's the sender is the presynaptic. So it's the first one. The presynaptic neuron and the postsynaptic neuron. Okay. Next one. Neuroglia that provide a, okay, protective coat around the cell bodies of peripheral neurons. Okay, let's look it up. I think that's probably that's either Schwann cells or satellite, but let's read it and see. Neuroglia that provide, okay, keep going because I need to read about I need to find what they're talking about with satellite cells. Okay, satellite cells produce a protective coat around the cell bodies of peripheral neurons. Oh, I see why I was getting confused. Okay, so look, Schwann cells produce the myelin sheaths that surround the axons of peripheral nerves. So Schwann cells produce the coating in peripheral nerves. Satellite cells actually coat the cell body. So not the sheath itself. The Schwann cells produce the myelin sheath, but satellite cells provide a protective coat around the cell bodies. So satellite cells. So go to the question. Satellite, Satellite, satellite cell. No, you have an extra letter in there. Satellite. So you need to remove, there you go. There you go. Which chemicals are essential for synaptic transmission? I think that would be neurotransmitters, but let's read it. That sounds like the most logical. Okay, keep going. Okay. Keep going. Chemicals called neurotransmitters carry out synaptic transmissions. Yes. So neurotransmitters. The distal ends of axons have one or more extensions called synaptic knobs, which contain many membrane sacs called synaptic vesicles. Dendrites do not have synaptic knobs. So neurotransmitters are the chemicals that you need to release. Mm-hmm. Conduction of nerve impulses is faster in unmyelinated neurons than in, no, that's reversed. It's faster in myelinated neurons. It's faster when it's coated, so that's false. What happens after an impulse reaches the synaptic knob of a presynaptic neuron? Okay, let's look it up. After the impulse reaches the synaptic knob. So, okay, go down a little bit. Okay, so the distal ends of axons have one or more extensions called synaptic knobs, which contain many membranous sacs called synaptic vesicles. When an impulse reaches the synaptic knob of a presynaptic neuron, some of the synaptic vesicles release neurotransmitter molecules by exocytosis. The neurotransmitter molecules diffuse across the synaptic cleft and react with specific receptors on the membrane of the postsynaptic cell. Okay, so let me see if I can find. I kind of understood that, I think. But let me see if I can find a thing on. Let me see. Where is my? Oh. Okay, hold on. Synaptic. Okay, let's see if this helps, because it might. Let me go and first of all, let me share that. So neurotransmitters are released into the synaptic cleft, I would say, from what we saw there. The speed of impulse conduction is proportional to the, I think that's diameter, but let's read it and make sure. I think it's the diameter. Okay, scroll down. I think it's underneath the picture. Okay, that's about, keep going. Let's go. The speed of impulse conduction is proportional to the diameter of the axon. The greater the diameter, the faster the impulse. So diameter is your answer there. Yeah. Inhibitory neurotransmitters may have the action of causing what? I don't know. we haven't gotten to this yet. So let's read it and see what happens. Inhibitory neurotransmitters. Let's read about the concept first. Neurotransmitters are not always excitatory. Often they will inhibit a neuron. An inhibitory stimulus will cause chlorine negative ions to flow in or potassium positive ions to flow out, making the charge more negative. This makes the neuron less likely to get excited. So chlorine negative in or potassium up. So let's look at your question. So negative ion in or potassium to flow out. So the first one or the third one and the third one. So there's two answers there. Okay. There we go. A junction between two neurons is a synapse. That's a synapse when you have, right. What is synthesized in the cytoplasm of the synaptic knob and stored in synaptic vesicles? Those are neurotransmitters. Neurotransmitters are synthesized in the cytoplasm and stored in the synaptic vesicles, so they can be released when the vesicles break open. Okay. Okay, in this schematic representation of a synapse, letter A is the blank and letter B is the blank. Okay, so letter A would be the presynaptic neuron. Uh-huh. And letter B would be the postsynaptic neuron. And we know that because of the way the arrow is pointing. So the impulse is traveling downward toward the picture. So it means it's leaving A, which means A is sending it and B would be receiving it. If it were going the other way, they would be reversed, I think. Okay, go ahead and let's. Okay, click and drag the elements in order. Place the following events in order that result in the release of a neurotransmitter. Okay, so okay, hold on. So then, okay, place the following events in order that result in the release of a neurotransmitter. So let's see. Okay, so here we have, let me see. A nerve cell arrives at a synaptic knob. I'm sorry, a nerve signal arrives at the knob. Voltage gated calcium channels open. Calcium enters the cell. ACH is released and diffuses across the cleft. Sodium channels open and sodium enters the cell, and a postsynaptic potential is produced. So this is going to be let's look it up and see if it tells me, because there's got a few things on there that I don't recognize. Okay. Most neurotransmitter molecules are synthesized in the cytoplasm of the synaptic knobs and stored in the synaptic vesicles. Okay. Okay, but I need to know. Okay, scroll down to the table of neurotransmitters and actions. Let's see. No, that's the names of them. That's the names of the, okay, keep going. When an action potential reaches the membrane of a synaptic knob, It increases the membrane's permeability to calcium ions by opening calcium channels in the membrane. So the action potential goes to the synaptic knob. The calcium ions, the calcium channels open. Calcium ions come in. Seminal vesicles fuse with the membrane. The neurotransmitter molecules. Okay, so the action put, okay, so this is what we're organizing. Table 9.3 is what we're organizing in the question. So scroll up so I can see it. Okay, so the action potential passes along an axon and over the surface of the synaptic knob. The synaptic knob becomes more permeable, calcium. So the calcium gates open, the calcium diffuses inward. The synaptic knob, or the synaptic vesicles, I should say, fuse to the synaptic knob. And then the synaptic vesicles release the neurotransmitter. Okay. So let's go to the question. Let's see what we can see. So the first thing would be the action potential reaches the synaptic knob. That's first. So the last one goes on top. Then let's see. Then let me check something. I forget when the synaptic I forget when the synaptic it's interfered in a second. I'm trying to close it, but it's not closing it. Okay. Then I think it's calcium channels open after that. And then calcium diffuses into the synaptic knob. Let me look at the order again. Let me look at the table, but I think that's the table that we were looking at. So action, okay. Synaptic knob, okay. So yeah. So calcium gates open. They come in. The synaptic vesicles fuse to the membrane, and then the synaptic vesicles release their neurotransmitter. Okay, let's go back. I think it's right. Let's go back. Calcium channels open. Yeah, because you need calcium for the synaptic vesicles to fuse and open. I believe that's correct. No. I believe that's right. Yeah. Okay. Next question. The spheres within the circle represent blank. Chemicals that allow for one-way communication between. I would think that would be neurotransmitters, but let's look at the book and make sure. But I think it is. I think that's what they're made to represent. Let's look at the diagram. We have to look at the diagram and see what the little round blue things are. So, okay, so these are, yeah, the little blue balls are neurotransmitters. And that's what yeah, so it's neurotransmitter. It's neurotransmitters. Uh-huh. Yeah, I thought that's what they were supposed to be based on what I was looking at. The brain is composed of blank neurons. It also contains neuroglia, which are blank then neurons. Okay, I don't know how many neurons we have, so we have to read it. I don't know what the number is. The brain is structured in reverse fashion to the spinal cord. The gray matter is on the outside, surrounding the deeper white matter. It is composed of about 100 billion multipolar neurons, which communicate with one another and with neurons in other parts of the nervous system. The brain also includes neuroglia, which outnumber the neurons. So it's 100 billion, and the neuroglia are more. Okay, so let's go to the question. It's 100 billion and greater, so it's the third one. Okay. Next question. When an impulse reaches the synaptic knob of a presynaptic neuron, okay, some of the synaptic vesicles release blank molecules that diffuse Okay, let's read it because I don't know. They want specific words there. So when keep going, it's underneath that. When keep going. When an impulse reaches, okay, so this is the sentence that it's referring to. When an impulse reaches the synaptic knob of a presynaptic neuron, some of the vesicles release neurotransmitter molecules by exocytosis. The neurotransmitter molecules diffuse across the synaptic cleft and react with specific receptors on the molecules. Okay, so neurotransmitter molecules okay, so let's go to the question. Neurotransmitter molecules is the first one. That diffuse across the synaptic cleft and react with specific. Okay, let's go back to the book again. Specific blank on the membrane. Go under the clinical application green part. React with specific receptors on the membrane. So they want receptors there of the postsynaptic cell. So go to the, it's receptors is the next part of it. Okay. Okay. Okay, the four major divisions of the brain. Okay, I don't know this, so I have to read it because we haven't looked at it yet. The brain is structured in, okay, we read that. The brain can be divided into four major portions. The cerebrum, the diencephalon, the brainstem, and the cerebellum. Cerebrum, diencephalon, brainstem, and cerebellum. Okay, so let's go to the questions. So the cerebrum, the diencephalon, the brainstem, and the cerebellum. Good job. An inhibitory neurotransmitter will make the postsynaptic membrane less likely to reach threshold level so that an action potential is less likely to occur. I think so. I think that's true. I think that's what inhibitory neurotransmitters mean. Because inhibitory neurotransmitters make excitation less likely. So I think that's a true statement. The blank is responsible for higher order functions such as reasoning, and the blank is responsible for coordination of voluntary muscle control as well as balance and posture. Okay, this is about brain parts. So we haven't read it. So we need to read about it. We need to read it. Okay, that's hemisphere dominance. That's cerebral cortex injuries. That's motor areas, association areas. I think it's probably gonna be, okay, here's the lobes. This is the lobes of the brain. The cerebrum is essentially, okay. Yeah, but what does it do? Okay, is that the beginning of this section or not? because they're talking about, okay, let's see. The cerebrum, the largest part, includes centers associated with sensory and motor functions and includes higher mental functions, that's the cerebrum. The diencephalon also produces sensory information. Neural pathways in the brain stem connect parts of the nervous system and relate visceral activities. And the cerebellum coordinate voluntary muscle movement. Okay, so the cerebrum is the one that helps you with your sensory and motor function and higher mental function. That's the cerebrum. And the cerebellum is the voluntary muscle. Okay, so let's go back. So the cerebrum, I think it's the last one. The cerebrum is responsible for higher order functions such as reasoning, and the cerebellum is responsible for coordination of voluntary muscle control, as well as balance and posture. Okay, so let's try that. Okay, next question. Where are neurotransmitters stored? In synaptic vesicles. I'm pretty sure they're in synaptic vesicles, but let's read about it. I think it's in synaptic vesicles from what I remember. Neurotransmitters, blah, blah, and are stored in the synaptic vesicles. Synthesized in the cytoplasm and stored in the synaptic vesicles, the little sacs that we saw, the little enclosures. So they're stored in synaptic vesicles. Okay. Cool. Next question. When an action potential reaches the membrane of a synaptic knob, it increases the membrane's permeability to when an action potential reaches the membrane. I want to say calcium, but I don't know for sure. Go ahead and read about it. I want to say it's calcium, but okay, go down below the table. When an action potential reaches, it increases the membrane's permeability to calcium ions by opening calcium ion channels in the membrane. Yeah, that's calcium ions. Calcium ions diffuse inward. So it's calcium. Go to the question. So calcium ions. Okay. In the brain, the blank matter is deep to the blank matter. Let's read that again because I think it's worded different. Okay, I think that's after. Let's scroll down under the yellow part because it was talking about how it's organized differently from the way the spinal cord works. Go back up to the brain again, the picture, because I want to see where the gray matter and the white matter is, but that's not showing me that. Okay. Okay. But I need, is there another picture of the white matter and the gray matter and the, scroll up a little bit. I'm looking at the, let's see. No, it doesn't say. Okay, scroll down. I don't see which one is. Okay, hold on. In the brain, let me see if I can look it up. Because I remember seeing it, but I don't remember. In the brain, the blank matter is deep to. Let's see what it says. So the white matter is deeper in the brain than the which is what I thought they said. The white, so go back to the question. The white matter is deep to the gray matter. It's a weirdly worded question, but I think that's what they mean. Because the gray matter's on the outside. Okay, let's see. Okay, yeah. The brain can be divided into the cerebrum. No, see, BRUM. M, not M, cerebrum. And the cerebellum, CEREBELLUM, cerebellum. Okay. Now we have to match the functions, okay? So provides conscious motor control, higher mental functions and reasoning. That's the cerebrum. That's the first one on top. The cerebrum. Processes sensory information is the diencephalon. Regulates visceral activities is the brainstem. So the one on the right goes in the third, yeah. And then cerebellum coordinates voluntary muscle movements and balance. Cool. Not too bad. Okay, I think we're good to go. You're pretty good. You don't have it's not due till the 23rd of March, so you're okay. So yeah, you don't have another chapter due until April, but you're just practicing for your midterm
Yeah.
on the 23rd. The lab midterm.
Yeah, because this one, yeah, I don't think it's like really homework. It's just like practice.
Yeah, she didn't give you, it says no points, just practice for your midterm. So, okay.
Okay.
I set up the receipts yesterday. And I'll be sending them out on Monday. I didn't want to send them on the weekend. Okay.
Okay.
Are you gonna be using all your hours? Because you still have 60 hours and you only used 18 this last month. So I don't know, are you going to be asking for fewer hours, or how do you think that's gonna work?
Well, I think, yeah, I'm gonna still use all the hours.
Use all, okay. Okay. All right. All right, Daniela. Well, then I will see you whenever I see you. I hope you're doing well, and I hope that this is making a little more sense to you than it was before. I'll talk to you soon, okay?
Okay, thank you.
You're welcome. Bye.